Hi Stats class, it's Mrs. Metzger. We're going to try to pick up where we left off last week when we were in class. Hopefully you'll have your packet at home with you along with your iPad so you can do calculations. If at any point you want to stop the video, you can stop it so that you can catch up on note taking. I've tried to incorporate the example throughout the process, so again, you could stop the video if you want to try some of the example things on your own before we go through them as well. Just know that we were anywhere from 10 to 12 days ahead of last year's group. So even though we didn't have class last week, we are not behind in any way, and we're going to keep moving forward. When we had class last week, we were talking about how do you analyze categorical variables. We said when you write up your null hypothesis, basically you're saying there's no association. We're going to write this phrase in words, and you're going to write it in context of the problem. Our problem last week was looking at gender versus lingering fright symptoms from being exposed to horror movies at a young age. The alternative says there is an association between the categorical variables. So in this case, gender and the ongoing fright symptoms. In order to tell if there is truly something going on between the two categorical variables, we have to calculate an expected cell count. And this is what we would expect to see in each of those joint cells. We're going to have a table set up. You're going to organize your data based on the results you received when you collected your raw information. We call this an R by C table, row times column. So we're looking at the observed counts that we collected with the expected cell counts that we're going to calculate. And this is the formula that we would use to calculate expected cell count. This formula is on your reference sheet. You don't have to memorize it, we just need to know how to apply it. So to calculate expected cell count, it's row total times column total divided by n, which is our total observed count. So here's the collected data for the ongoing fright symptoms case. We had 117 people participate. So to calculate the expected count here for female who do not experience lingering fright symptoms, we're going to take our row total in blue, 81, times our column total in blue, 79, and divide it by 117. So we are expecting to see 54.6923 values here. This is not that different from what we actually observed with 50. We're going to do this for each of our cells. So now males that do not experience lingering fright symptoms, row total in blue times column total in blue divided by 117. So we're expecting to see about 26.3077. And I usually leave it unrounded. I do not make it a whole person. These are not actual people. They're just calculated values. So we'll do this for females who do have lingering fright symptoms. Row total, 36 times column total, 79 divided by 117. And then finally, the males that do experience lingering fright symptoms. We only have to do it for the joint cells. We don't have to do it for any of the marginal totals. So what we have essentially here is a 2 by 2 table. What we'll do to test our null hypothesis is we're going to run an inference test that compares what we've observed with what we've calculated in our expected cell counts. This is called the chi-square statistic, and it's looking at how far the observed counts are from what we've calculated in the expected counts. So this formula is also on your reference sheet. This is our Greek letter chi, and it is squared. We're going to sum the observed count minus the expected count squared all over the expected count. And we'll go through this procedure in the upcoming example. This is the procedure we'll use to calculate our chi-square test statistic. So what you're going to do is calculate all the expected cell counts, and then we're going to find the difference between observed and expected and square it. And that's what you're seeing in the numerator in the formula. And then we'll divide each squared difference by the expected count. That's what you're seeing in the denominator. And then we'll add up all of our results. The larger the number, the further into the right-hand side of the tail we're going to be, and the more evidence against the null we're going to have. So your chi-square distribution is pretty skewed. You can see that what we're looking for is, does our value lie in this pink shaded area? And again, the further away you are from 0, the more evidence you're going to have against the null. So we're always in the upper tail of the distribution. Using the chi-square test statistic, we do use degrees of freedom. So the more cells we have, the more accurate our distribution will become. You can see if we calculate degrees of freedom at 1, this is an exponential decay. This is very skewed. Degrees of freedom of 4 here at the red curve, still skewed, but not as badly. And by the time we get to degrees of freedom of 8, the curve is starting to normalize. So the more information that you have, the more accurate and more normal your distribution will become. So if the expected counts that you calculate and the observed counts you gathered are very different, it's going to start to increase the value of chi-square. So it's going to become larger and larger, and each cell will play a role in this. As you start to see values accruing in the calculation, large values of chi-square are going to start to provide strong evidence against your null hypothesis. Remember, it doesn't mean that the alternative is true, it's just we have evidence against the null. We're going to use the calculator. There is a chi-square CDF function. When you go into distributions, you'll just have to scroll for it. You could also use table F from our book, but you need to know the number of degrees of freedom that are present for your particular table. So to calculate degrees of freedom, it's row minus 1, column minus 1, and then you want to multiply those two values together. In order to be able to use your chi-square test statistic, your average expected cell count must be 5 or more, and all individual expected counts have to be 1 or greater. So again, the more data you gather, the more likely this is going to occur. The only difference is when we use a 2x2 table, which is what we were using for lingering fright symptoms and gender, all 4 expected cell counts should be 5 or more, and that just accounts for smaller amounts of data that we have. All right, so let's compute the chi-square test statistic and the p-value for fright symptoms versus gender. So again, on your iPad, you're going to go to menu, and this is the chi-square CDF. So we'll need this at the very end once we obtain our chi-square value. So here's what we want to do. Chi-square is calculating. You're going to find the sum of all these expected differences. So this was our first cell. We observed 50. We're going to subtract our expected cell count. We're going to square it and divide by expected cell count. So we're going to do this for each cell. Here's the next cell value we calculated, observed minus expected, squared over expected. The next result, and the fourth result. So each of these pieces can be converted to a decimal equivalent. There's the yellow calculation, the red calculation, the pink calculation, and the blue. So we're going to add all of these up, and we have a chi-square value of 4.02839. Not huge, but still it might be far enough away from 0 to show that we have some evidence. Now, I neglected to include this when preparing the slideshow. But we do need to calculate our p-value at this point. So on the calculator, you would go to distributions, and you're going to select chi-square. The lower bound is always going to be 0. You can see that on the previous slide on that curve. The upper bound is the chi-square statistic we just calculated, 4.02839. And degrees of freedom, we had a 2 by 2 table. So 2 minus 1 is 1. The column 2 minus 1 is also 1, and 1 times 1 is only 1. We only have 1 degree of freedom. We are in the upper tail, so it's always going to be a 1 minus, but the p-value is 0.044743. So typically if alpha is 0.05, we would have significant evidence at this point to accept our alternative hypothesis that there is some kind of association between gender and associated fright symptoms. The students at Syracuse do use Minitab quite often. This is a Minitab output calculating chi-square statistics. What you can see, they're comparing gender and smoking cigarettes. Down here, the cell contents describe what's being shown. So the top row is the count, which was the actual data observed. The second is the percentage of the row. So if you look at female, 94.49 said no, they don't smoke. 5.51 said yes, they do smoke. That's 100% for that particular category. The third row is the expected cell count. So they expected to see 117.45. They actually observed 120. That is not that different. And for yeses, they observed 7, and they expected 9.55. Again, not that different. But all of these cells are set up in the same way, and they talk about how they contribute individually to the chi-square test statistic. Down here at the bottom, they have the chi-square value, the degrees of freedom, because this is a 2 by 2 table, and then the associated p-value. So when we set this table up, typically at the start of the study, we're going under the assumption that there probably is some kind of connection between the two categorical variables. And even though we did see some results, they don't know if the gender is the cause or the fright symptoms are the cause. We don't really know who's causing what. We just know that there's an association. Unfortunately, with the table, you can experience confounding variables. So there might be some third or fourth variable playing a role in our results, and it's impossible to tell what that is. Just know that that is something that might be hanging around. And typically, since we're not comparing those other variables, we're not going to be able to investigate them. So the testing really stops there. So the chi-square test will allow us to either have evidence against the null or evidence in favor of our alternative hypothesis, and it will show us that there is a relationship between the variables, but you would need to investigate further to draw any kind of conclusions. If you can see that there's evidence against the null hypothesis, then the best way to show the distinction between the variables or the cause and effect is to calculate the conditional distributions and look at percentages. And typically we do this before we even begin. So the chi-square test statistic is just verifying what we've already seen in our table that displays our percentages. All right, we're going to stop there. Typically we cover 8 to 10 slides every class, and that's what I'll try to stick with. This is the homework problem associated with this lesson. I did put a picture of it here in case you don't have your textbook at home. If you have questions while you're going through homework or any of the notes that you've taken, please feel free to call me at home. I've listed my home number here. call between 8 and 3, which are normal school hours. You can really text me anytime, or you can shoot me an email and let me know that you're struggling or you have a question, or even if you just want to chat. Have a great day.